as designer and the web developer. I will be giving overview of GlyGen in the portal part. Let me introduce you to GlyGen. GlyGen is an international project funded by NIH to facilitate glycoscience research. GlyGen retrieves information from multiple international data sources and integrates this data into one source, GlyGen. GlyGen allows you to perform unique searches that cannot be executed in any other existing databases alone. This is our international team from multiple institutions in US and international institutions. During our presentations, if you will have a questions or you want to give us suggestions, you can do by multiple ways. First, you can submit your question in a via WebEx chart, which located on the right bottom corner. Also, you can go on our website. We have navigate to contact us page from navigation bar on the top or link on every page on a footer. You can submit your question. We can get to you as soon as possible. Or also in some of the pages, in the multiple pages, we also have a feedback gadget where you can submit your questions or suggestions. This is where you can contact us. Right, your feedback will help us improve our website. You can find feedback, leave us feedback from a navigation bar on the top bar, from drop down menu, or on the home page on the right side. You just need to fill out answer a few questions, and will help us improve our website. Let me give you a little overview of our website. We provide you with multiple searches: quick search, try-me, global, GlyGen search, composition search, protein, glycoprotein. Also on our website, you can learn more about us, about our team, what our vision and mission. You can reach out with us via contact us page or leave us feedback. On the home page, you can see our updated versions of the data or portal. You can read our news from a homepage, and we will let you know how to properly cite our website when you're using for your researches. Also, we can give you, you can see what kind of data we have, and we provide you with database-based statistics in homepage. If you're new to our website or if you're new to research in glycobiology, we are providing tutorials for you. We also give you resources where you can learn more about this field or give you more tools. We're giving to you access to our media for you to use, like presentations, and we telling you what kind of frameworks or libraries we're using to develop our website. Right, this is how our home page looks like. On the right side, you can see our database statistics, and we have a multiple species: Homo, Mars, and Ratrus. Also, we have SARS, CoV-1, and CoV-2. Right, on the top bar, you can get access to our multiple searches from a home page. On navigation bar, on the top navigation bar, or by clicking one of these images or one of these queries in a quick search in the try-me. Let's first of all, I want to show you a quick search where you are searching over here by your queries. In the quick search, you can do multiple searches by GlyGen, by protein, by species, or by disease. All you have to do, just insert your ID. And in this example, we're searching query. It's asking what are enzymes involved in biosynthesis of glycan X, which we will put ID in human. So we will searching for protein, and here's a summary of your protein. By selecting uniprot accession on the left side, or this table, let me tell you more about this table. This is list table. Well, it gives you gene name, uniprot, KB name, organism, RefSeq name, or RefSeq accession. You can also filter this table. Let's take a look example, selected example. Select uniprot KB accession, and it will take you to details of that protein. Later, I will give you in the details overview of this page. Also, try me search, and this try me search, you searching by query, and we already inserted ID for you. All you have to do, just click a question, um, provides example will provide it for you, and it takes it's doing the search for you. And by selected Uniprot KPI session on the left side, it will take you to details, so you can view details of that protein. Right, in other search we have a global search. You can navigate from a header, from any page on a header. Let's select, let's search L13. It will take you to very well organized global search page where you can see details. In the blue color you can click, it will take you to different page. I will show you in a minute. We will select number 3 in genes. And we will see 3 genes on this page. Let's select Uniprot KBS session of my interest. It will take us to detail page. Right, I won't give you little emphasis over here on the general section. When you select RefSeq accession number, it will take you to NCBI section. On the right, this is where you can read additional information related to your search, to your protein. Right, on the right side, you have to navigate to link out sources, find GlyGen. By clicking that GlyGen link, it will take you back to details of your proteins. This is where you just came from. Right, I want to quickly show you Glycan search. You can click one of these images or select from Explore on the top. It will take you to the simple search. Simple search, it's you searching by using simple search when you know what you're searching for. It's like a shortcut for you. By selecting any ideas, it will take you to the list page. When you will select idea of your search, it will take you to detail page. All right. So next, we will search advanced search. I will show you. You can search, select multiple inputs over here on the left side. Let's say glycan ID, monoisotopic mass, number of sugars, organism, glycan type, glycosylated proteins, glycan motifs, bison synthetic enzymes, and a PubMed ID. And you can select multiple selection or inputs, and it will filter your search. In my case, I will search just by glycan ID. Click search. It will take me to list. Here I can see additional information of my glycan on the table. And then it will take me to glycan detail page. We also have a tools. Let me go back over here, icon on the top, related glycans. By clicking on related glycans, it will take you to this page where you can see related glycans. The same what name said, glycans to you glycan. Right, we have another tool, it's a motif. Over here, by selecting one of this motif, it will take you to motif page where you can see additional information, glycans containing this motif. All right, in other search, we have a composition search. By selecting residues and a filter by using min and max numbers and contains maybe yes or no, I selected these two and said yes. By clicking search, it will take you to summary of your selection, glycan search. Select one of these glycogen accession numbers. It will take you to details of that idea. And a very similar, all glycans have a similar detail pages, like a sections. I meant to say sections. but it has individual data for each ID. Right. Also, we have tutorials for you. If you're new to this field or you're new to our GlyGen, we provided tutorials for you. Just read and listen. It will be very helpful, and let us know if it's helpful. Also, we have please give us a credit, cite us on your research papers, give us feedback. Let me take you to our how to cite page. We provided this page for you. This is how you will cite. We give it to you example how to cite us. This is our papers, and here's the website citation. This is how we will cite us as a website. Right, let me give you a live demo of our searches and our homepage. This is our homepage. Over here, you can perform multiple searches, like I mentioned before, glycan protein, glycoprotein. Here's a global search. You also click one of these icons, images. Here's a quick search. Just click on one of these questions and you will start performing search. On a try me, just click on this example. It will take you to the search. Right, let me give you a detailed overview of glycoprotein search. You will land on a simple search. Over here again, you can search any category by selecting your ID. It also will include context, um, including the context match. By selecting one of these diseases, you will just search, just put your disease and search. Right, I want to give emphasis on advanced search. In advanced search, we have a multiple input for selection. It includes UniProt KB accession, RevSec accession, chemical mass, organism, protein name, gene name, go ID, interacting glycan, glycosylated amino acid, protein or peptide sequence, pathway ID, PubMed ID, glycosylation evidence type. You can select multiple of these, filter your search by selecting multiple IDs. In my case, I want to select, we provided examples for you if you're new to our website, or you can explore more about what UniProt KB means. Also, we have, when you mouse over on a question, we have more details to this UniProt accession number. Also, we created a cool feature by clicking this ID, it will input it for you. It's example. Let's search. And by clicking this number, I want to show you something else. On a summary of your glycoprotein search, if you change your mind and you want to change your search, you just have to click Modify Search. It will take you back to your advanced search. In my case, I don't want to change anything. I just will continue with this. By clicking UniProt accession, it will take me to details of this glycoprotein accession. I want to give you emphasis of a few tools. Like I mentioned before, by selecting RevSec accession, it will take you to NCBI website where you can read more additional information about that. And on the right side, I'm looking for linked out to external resources GlyGen. By clicking that, you go back to details for the protein. This is network we created for you. Species, function, go annotation, glycosylation. Okay, I want to show you one search that is available only in the GlyGen. Nowhere else is available. Okay, this is unique search. And we will search on what other proteins might have glycans interested you. By clicking glycogen accession, you will see details of that glycan. And this is, we have a couple tools which Rakhine, our new speaker, she will go over the details and my TIFF page. And here, let's see, found glycoproteins by synthesis, digital sequences, cross-references, and publications related to that glycan ID. Right. On the found glycoproteins, if I will select one of these UniProt KBS session number, it will take me back where I just came from. In the glycosylation section, we have with reported glycans or without reported glycans, which we can see residue over here. I will show you in a minute. And also we can see residue sites over here as well. Let me click on this link. This is another tool we provided for you. This is a site view and table of this site view. You can see site annotation. Right, also you can change the sequence view. By changing sequence view, you will see different site annotation. This is how it works. All right. It also has similar information like a RefSec accession. Right. Let me go back to details of the protein. In another section, we have another interesting tool I want to show you is sequences. It's ProtVista. By selecting this and glycan sites or glycan sites, you see the highlights. Let's go to ProtVista. I guess maybe, I don't know, my internet is slow. Let me try again. All right, go back. So I want to show you very interesting glycosylation. We have N-glycan site, O-glycan site, or SIG1. By mouse over, you can see image of this N-glycan or additional information. Also, by, high, by, by mouse over one of these names, it will highlight your where is it located. All right, go back to the protein we just came from. So it also gives you information about pathways. In other isoform, we have interesting tools for you. You can show or hide the sequences, and let's see alignment of that isoform. Isoform alignment for protein with this protein. And this is alignment summary. All right, let's go back to protein details and observe more. Here's the homologs. You can select homologs or OMA or M guide. Also have alignment here, show sequences. I have to select it first to see alignment. Right. Here's another table mutation where you can see information of your mutation, expression tissue, expression disease, cross references, and publications related to that protein. All right. Also, you can download these details in a multiple formats, FASTA or JSON. Let me briefly give you overview of other searches we have. This is already mentioned, quick search. By selecting this, it will take you to a quick search page where you can search by queries, just insert your ID, search by protein, search by species, search by, let's say, other species, search by disease. This is how it looks. So I want to give you example of search of this query. What are enzymes involved in biosynthesis of glycan, of this glycan in human? It will take us to the list page where we can select our ID, and it will take us details for this protein. And it's very similar what I just was showing you, only it shows you specific details for this specific ID. Right. Go back to home page. Try me. This you can find on navigation bar on the top. Over here on Try me section. Let's select one of these questions, and I do already pre-filled for you. By selecting Uniprot accession on the left side, let me go back. I think I want to select this one, Unamodium. And I want to show you details of that ID. Right. So we covered Glycoprotein search, quick search, try me. Let me show you how it works global search. By importing one of interested your ID, click enter or search. It will take you to this unique page where you can see additional information about the search. In this example, I want to select gene. It has number 3. By selecting one of these, you import a session. It will take you to detail for that protein. And you know the drill, detail page. Right, one more thing I want to show you. Last more question, search, GlyGen search. So see, you landed on a simple search. You just select your category or search by any category. Advanced search, where you can select multiple searches or filter by type or by organism. Select your organism, and it gives you a subtype. This is how you filter. Just click search. It will take you to the list page, summary of that search. Right. On the GlyGen search, also you can find composition search. You can select yes, or you can select no. If you didn't select it, will maybe. Or you can also change minimum and maximum numbers. Select. Also, you can select all no, or all yes, or clear fields, undo, redo, and then hit search GlyGen. It will take you to the list page with a summary of your search. Also, we have tutorials for new users. If you're new in this field, or our website, GlyGen tool, so we provide tutorials for you. Very easy. If you still have questions or you want to give us suggestion, we have a feedback gadget over here on the right side. It's on multiple pages. You can find that. Just submit your suggestion or questions, and we will get back to you very soon. Another place you can find us, contact us here, or on every page on the footer, contact us. Just leave us a message, stay in touch, and we will get back to you as soon as possible. And the last more thing, how to cite. When you use our website, please use our website, where you will make us very happy if you use our website for your research. And this is how you will cite. Give us a credit. Also, you can, this is example of how to cite our website. All right, I think I covered my portion, and I want, let me introduce you to our database specialist, Rakhine. She will give you an overview of our database. Rakhine?
Great, thank you, Tatiana.
Stop
Sharing my screen. Hi everyone, I'm Rakhine and I work on GlyGen as part of the data management team. Today I'll be giving a brief description of the GlyGen data, our resources, followed by a live demo of our website. Just as a reminder, if you have any questions during the presentation, please add them in the chat box, and if we have enough time, we'll be happy to answer them one by one. So here is the basic overview of how GlyGen data looks like. You can think of it as consisting of two main entities, protein and glycan. And then you can divide each of these entities further into important pieces of information, such as protein function, glycan mass, glycan classification, protein expression, etc. Currently, GlyGen has glycan and protein data for different species that we have added over the last 3 years. We started with human, and then we expanded to mouse and rat. And this year, we have added hepatitis C virus as well as SARS-1 and SARS-2 data. In the upcoming years, we are also adding additional species. So how does GlyGen generate this data? You can think of GlyGen as a super collaborative environment where we connect with major resources, which could be protein-centric, such as UniProt or PDB, or glycan-centric, such as Glycogen, PubChem, STEBI, or glycoprotein-centric, such as Unicar KB or Glyconnect. With our collaborative efforts, we collect data from these resources. We implement quality control techniques as well as generate rules and algorithms to filter and add annotations to provide value-added information to our users. This way, a user can perform a search across multiple domains. As you can see on the slide, a user can easily generate a search where he or she can look for a protein implicated in, let's say, disease X, and then can study what type of glycans are found on that protein. Further, the user can filter those glycans to a specific classification type, let's say N or O glycan, or even search for a glycan for a particular mass reported on that protein. As such, there are several other such use cases that a user could do to get their desired results. For the purpose of this demonstration, I'll be going through one such use case. I'll be shifting over to the browser. Okay. So as Tatiana explained, this is our main GlyGen homepage. And for today's demo, I'll be using the GlyGen global search to search for any data in GlyGen for SARS. Now this will provide me all the entities, identifiers, or any values that are associated with SARS. And now it generated 3 different sections: glycan, protein, and glycoproteins. Now in glycoprotein section, I will go to the identifier section and click on it. Now this will take me to the GlyGen list page, where you can save this URL and maybe, let's say, return back to the same URL a week later and just click on update results. So if there are more information for that search, you will get the latest results. Or you could click on the modify search option, where you could add additional filters or change your query completely. Now I'm going to go to the spike glycoprotein expression for SARS. Now this will take you to the glycoprotein detail page, where you can find detailed information about that protein. For example, mass, function, GU annotation, and importantly, glycosylation. Now here you can see a table which provides you information on what site of that protein you can find what type of a glycan. Now the glycan can be represented in an image or an expression format. This way you can browse through the table and find all the glycans that have been reported by available publications through this section. Now, as you can see, the compositions that the paper reported, the glycans that the paper reported are just the composition. In places where the publication do report or define structure, we do capture that. Now, if you want to know more about where this information came from, you can use these evidence badges, which will take you to the publication section or the collaborator and understand the source of this information. Now, for example, if you want to explore more about this glycan, what you can do is from this table, click on this particular accession, which will take you to the detail page of that glycan. Now, this page is similar to the protein detail page I was showing you earlier, And now in this case, it will have all the information about that glycan, such as its accession, its image, mass, composition, any other species that has been found to be reported in, and it's the glycoprotein that it has been found. For example, the same composition that we saw in SARS was also reported on the IgHM protein at site 272. Now, as I mentioned earlier, the publication only reported the composition of a glycan. Now, if you want to study more about this particular glycan, or you want to find the related glycan, you can click on the top right badge over here called Related Glycan, which will take you to a GNOME browser. Now, this will show you different organization or structure of the glycan with the same monosaccharide composition. Here, the monosaccharide query has already been added. Now, for example, if you want to explore a bit more about this particular glycan, if you click on it, it will take you to a tree-like structure where if you move along the tree, it will keep giving you more defined structures as you move down the tree. Now, for example, if you are interested in this particular accession, it's fully defined. I can right-click on it. Either I can copy the accession or I can move to the GlyGen page for this particular accession. This way, I came from a composition to its related structures to back to the GlyGen page for this particular accession. Now, again, you can find the mass, composition, species, and motif as well. So if you want to study more about the particular motif this particular glycan has, you can click on its label, and it will redirect you to the motif page. It will redirect you to the motif detail page, where you can find if the same motif was found on any other glycans or not. Now, for instance, if you want to search for the same glycan in some other databases, then you can use the cross-references section on the page, where you can find different databases listed and its corresponding accessions. Now, if you want to search for the same glycan in PubChem compound pages, you can select for this accession, which will redirect you to the corresponding PubChem pages, and you can understand different aspects such as chemical and physical properties, spectral information, etc. Now, if you want to jump back to GlyGen, what you can do is go to the biologic description section. Here we have cross-linked from PubChem to GlyGen back. So you can click on this link, and it will take you back to the same page. Now, if you have a different glycan composition in mind that you want to query the GlyGen database against, what you could do is you could go to the Explore tab, go to GlyGen Search, and go under the Composition Search. Now here, I want to do a very specific search, wherein I want a glycan which has, let's go with 6 hexose sugars and 2 N-acetyl-hexamine. I'll click on Search Glycan, and it will return me back all the glycans that have that specific monosaccharide composition. Again, we come back to the list page, where you can either download the entire results of 201 glycans in a CSV or a JSON format, or if you want to download data for one particular of your interest glycan, you can find the download badges on all the detail pages on the top right corner, where you can download it in the JSON or in the PNG format. Another way to access GlyGen data is through this tab, which takes you to a dedicated page where we have presented all the GlyGen data that we integrated from different resources in simple tabular format, where you can either filter by species, by molecule, by file type, by protein, or you could directly search for the source of it, like the UniProt or something like that. So for example, if you want to, if you're interested in this first table, you can click on View Details. Now here you can find, you can either download this table in a CSV format, or click on a README, which will give you all the information such as who provided this information, what are the input files, what are the output files, etc. Additionally, this particular interface will also provide you with different versions associated with it. As we get more information or we develop different filter criteria, we update our GlyGen data. And with every update, you can find a different information. You can find the update information through the release history tab as well. Now, as I mentioned earlier, GlyGen is a super collaborative environment. So if you have data that you would like to contribute to GlyGen and integrate with us, you can use the upload file interface where you can download a simple CSV or a TXT format file. Now, this interface will also ask you to provide your email and your organization information. This way we can contact you and we can work together to integrate your data into GlyGen, as well as link your data back to your resource from the GlyGen protein and glycan pages. Additionally, you can also download all our data from the using our API and Sparkle services. You can also visit our GlyGen FAQ pages where we constantly update it based on the number of user queries we do. If you have any additional questions, you can always go to the GlyGen homepage where you can leave the feedback to us. This feedback can also be found on every GlyGen page in form of the small tab where you can report any problems, you can ask any questions you have or leave a suggestion, and we will get back to you. So thank you so much for listening to us and giving us an opportunity to present. If there are any questions in the chat box, we can answer that first. Or if you have any questions, please feel free to jump right in. If your question is not answered within the time, you can always use this feedback option, or you can go under help and contact us and leave us a message, and we will get back to you. Thank you. So I don't see any questions in the questions. So if anybody has a question right off the bat, please feel free to ask us. So this session is till 6, so we have time to answer any queries you would have. Thank you.
Is how can I get a list of so the question is how can I get a list of glycoproteins that have glycosylation on tyrosine residues? Is there a specific search to do such kind of searches?
Yeah, so let me just share my
Show
screen
that
again. So what you could do is you could go to the Explore tab and go to the glycoprotein search right first. And then you can move to the advanced search option. You can scroll down and use the tyrosine. G?
Yes, tyrosine.
Okay. And you can even filter for organism, let's say Homo sapiens. And even additional filters if you want to say reported, predicted, or both. You want to select both and then do search glycoprotein. This way you can come to the list page and again download results or save this result as I said.
All right. The other question that I've received is there any way to search for data sets that contain only the glycosylation sites or associated information in GlyGen?
Yes. So what you could do is, as I showed you before about the glycoproteins, that's one way to select it. You can filter it based on species. Second thing you could do is you could go on the data tab And you could use the search bar to filter out the tables that you can see. So if I just say glycosylation, so it will give you different tables. Like the first one is the human glycosylation sites that we collected from UniProt. And then you can also filter out the species that you would want to see. Something like that. So, or additional option is you can also use our API and Sparkle to modify the query as easily as you would like.
Right. Thanks, Roy. And the last question that I have is, what's the easy way to get a list of human glycosyl transferases and human glycohydrolases?
I can answer this question.
Sure, go ahead, Tatiana.
Yeah, I'm sharing. Right, you have to go to quick search, which locates it, you can find on the navigation bar, and you have to search by species. Here's what are glycosyltransferases in species X, or what are glycosylohydrolysis in species X. You just have to select organism human and go ahead search. This is you will get detailed summary of that protein and select of your UniProt KBS session number. Okay, let me go back, modify my search. And here is another one. You just select your species of your interest. You set your HOMA. You just click search. And you will see summary of your protein on your search. Selecting one of these protein, you have to choose one and you will see details of that. Another interesting thing I want to show you is the search by species. You what are reported or predicted glycosylated proteins and species. So you can also over here select human species and select type of it. It can be reported, predicted, or both. Let's try both. Filter your search this way, Homo sapiens in both. This is the result. Select one of your UniProt KBS session and you will see details of that. Did I answer your question?
Yes, thank you, Tatiana.
Right. Do we have more questions?
Oh, I don't see any questions in the chat box, so that was the last question.
All right, thank you so much. Thank you everyone for joining us and for listening us. I hope you benefited and learned a lot. Please, please, please use our GlyGen as a resource tool for your researches. Thank you everyone. I think this session is concluded. It's over.